Hi, my name is Brittany Toba, and in this video I will be explaining the topic of epigenetics. I will be using data and evidence from my research of scientific journal articles and reports to explain to you just how your grandmother's lifestyle can affect and influence your health. I will be explaining the dietary and dramatic environmental changes made in the early life of rodents and their consequences and the relationship between epigenetics and disease states such as cancer. DNA is a part of every living system on this planet. It accounts for the 25,000 genes in our bodies that were identified by the Human Genome Project. Genes are how we distinguish individuals and are basically our instruction book for life. The epigenome is the gene's instruction book, full of coding instructions, chemical markers, and switches that are found along the length of DNA's double helix that turn the gene on or off. The term epigenetics refers to the difference in gene expression that is able to be passed on from an individual's parent through cell division, mitosis and meiosis, without making any changes to the genetic sequence. For example, removing a letter from the sequence. In the past few years, scientists and researchers have begun to understand epigenetics more clearly than ever before. From understanding the molecular sequences and patterns that determine which genes can be turned on and off, to the discovery on how epigenetic signals experienced from the environment can be passed down several generations without the genetic sequence being altered. Research is still being conducted to understand the ways in which this switching on and off of genes and changes in genes occur. Some of these changes include DNA methylation, which involves modifying bases such as CpG dinucleotides by adding a methyl group, generally known as a carbon atom and 3 hydrogen atoms, that disrupts their transcription factor binding to gene sites, chemical signals that activate the gene, essentially silencing or switching off genes. Figure 1 shows this process. Post-translational and histone modifications. This process involves the histone code to be blocked. DNA in every cell is wound around proteins called histones. An acetyl group is added and loosens up the bond between DNA and histones, allowing it to be successfully transcribed. This acetyl group is blocked, leaving the DNA to be tightly wound around the histones due to their positive charge and DNA's negative charge, thus preventing successful transcription. This is shown in Figure 2. Non-coding RNAs. Small non-coding RNAs refer to a group of RNAs that lead to the deterioration of messenger RNA and restrain gene expression. Nucleotide microRNAs are part of this group, which are the result of primary RNAs being cleaved 4 times then transported into the nucleus, exerting their effect. DNA methylation was a change discovered in the offspring of agouti mice in the study by Girdle and Waterland. Agouti mice are fat yellow mice that carry the agouti gene, making the mice prone to cancers and diabetes. When the mice breed, their children carry this gene and are identical to their parents. However, in this experiment, a group of agouti mice gave birth to slender brown mice that did not receive the effects of the agouti gene. The erasing of this gene was achieved without the modification of a letter of the mice's DNA, but through the change of the mother's diet just before conception. The diet of these non-agouti birthing mice was high in methyl donor foods such as onions, garlic, beetroots, and food supplements often given to pregnant women. Through DNA methylation, the food's methyl molecules work their way into the embryo's chromosomes and onto the agouti gene, switching or silencing the gene's effects. Though these diets rich in methyl are proven to be a positive effect, no one yet knows where else the methyl donors may end up. Though it has a positive effect, it may have a negative effect on another. So if there are too many methyl donors being consumed, you may be overmethylating other genes. Another study was conducted on the effect of adverse conditions in early life rodents that showed can severely impact the child's developing brain with an increased susceptibility to mood disorders in adulthood. Developmental plasticity defines an organism's ability to adapt to the environment during early life and to implement long-lasting changes in key biological programs, such as the hypothalamo-pituitary adrenal gland (HPA) system, which is a major component of the body's stress response. In depressed patients affected by neglect or abuse, hyperactivity of the HPA is found. On exposure to a stressor, the body activates stress systems to prepare for events that may threaten well-being or survival. The nervous system activates a rapid short-lived response while the HPA axis is slower. The HPA axis is core to the long-term regulation of systems controlling stress responsively. After exposure to a stressor, neuropeptides, CRH, corticotrophin-releasing hormone, and AVO, arginine vasopressin, are released from the hypothalamus into the portal vessel system. The neuropeptides bind and activate specific receptors to synthesize and release glucocorticoid hormones, cortisol in primates and cortisone in rodents. Glucocorticoids mobilize glucose from energy stores and increase cardiovascular tone, constricting blood vessels. Clinical studies found that depression is characterized by hypothalamic overdrive of CRH or AVP systems in depressed individuals. Figure 3 shows a hypothalamo-pituitary adrenal stress axis. Studies in mice pups demonstrate that exposure to maternal separation for 3 hours per day for the first 2 weeks of life showed effects later on in life of stress hyperactivity, such as elevated corticosterone secretion and altered feedback inhibition of the HPA axis, reducing DNA methylation in the hypothalamus. Pups that received high level of maternal care sustained decreased hippocampal sensitivity to glucocorticoid hormones, decreased CRH levels, and reduced HPA axis responses to stress. This is because licking and grooming releases serotonin in the pup's brain, activating receptors that send proteins to turn on the gene that inhibits stress response. DNA hypomethylation has been found to associate with schizophrenia and bipolar disorder. Figure 4 shows how early life experience can persistently alter expression levels of key genes through epigenetic marking throughout later life. Figure 5 shows experience-dependent activation of neurons during early inhibiting MECP2 binding to and repressing the AVP gene. This favors DNA hypomethylation. Additional pathways and epigenetic targets on early life stress also exist. A study that showed rats reared in hostile postnatal environments exhibited reduced expression of brain-derived neurotrophic factor in adulthood. BDNF is a neurotrophin that regulates neurodevelopment, neuroplasticity, and neuronal functions. These results suggest that through epigenetic changes we may have inherited changes seen in our ancestors' life, and that these changes may be seen in humans. A study by Marcus Pembury aimed to investigate this theory in 2005, where 2 centuries of crop yield and food price records were drawn. He noted that the town's food supply may have had health effects spanning 2 generations. Their findings showed that grandfathers who lived their preteens during times of plenty were likely to have grandsons with diabetes, and affected the mortality rate of grandsons only. The same situation was seen with the grandmothers and granddaughters. Further analysis supported that the genes on sex-specific X and Y chromosomes were being affected by epigenetic signals. He found that timing was critical to the impact of these changes. The granddaughters most affected were those whose grandmothers experienced times of plenty while in utero or as infants. The grandsons were those whose grandfathers experienced plenty during the period just before adolescence. Although the individual's epigenome is important in early fetal development, scientists have discovered that the epigenome can change in response to the environment throughout their lifetime. These changes are important in the development and spread of cancer. Cancer cells have genome-wide abnormalities at the epigenetic level. These abnormalities give a selective growth advantage to neoplastic cells, leading to deficiencies, inhibited cell growth, and tumorigenicity, tumors. Figure 6 shows these mechanisms. Tumorigenesis is the result of the activation or inactivation of pathways. It was previously believed that tumours were the result from genetic mutations, amplifications, gene rearrangements, or deletions. Studies now show that DNA methylation is another way of silencing tumour suppressor genes, much like genetic mutations. Abnormal DNA methylation was more frequent than copy number changes when studied on a genome level in malignant gliomas and colorectal cancer. DNA methylation effects on pathways can be either direct by affecting promoters of tumour suppressor genes, or indirect by silencing inhibitor of oncogenes, a gene that has the potential to cause cancer. Methylation is an early event in a tumour, but plays a major role in tumour growth and the accumulation of genetic and epigenetic abnormalities. Hypomethylation can also reactivate normally silenced genes that could potentially affect normal gene expression and potential activation of growth-promoting pathways. There is limited information on histone modification in cancer cells and tumours. Studies have shown a loss of specific histones can be associated with cancer. Transition and histone modifications are characterized as a two-way street controlled by a balance of catalytic, rate-changing, enzymes. Disrupting this balance can occur through altered expression or function of epigenetic changes. This is found to affect both murine and in-human neoplasia. If the histone's methyltransferase PCG protein catalyzes trimethylation, its overexpression has been found to create tumour growth in melanomas, lymphomas, and prostate and breast cancers. The reason why histone modification is limited is due to the fact that it requires a large number of fresh or frozen cells. Studies confirm that miRNA modifications lead to things such as defective miRNA and impaired cellular differentiation, suggesting that miRNAs may be associated in human neoplasma, abnormal growth of cells. Through the study of epigenetics in cancer cells, researchers have made two classes of drugs that can modify epigenetics and have been approved. These drugs can work because epigenetic changes are potentially irreversible, whereas genetic changes such as mutations are not. These drugs are DNA methylation inhibitors and HDAC inhibitors. These are shown in Table 1. DNA methylation inhibitors are nucleosides, simple bases, analogues, similar molecular structures that exert their demethylating activity through an irreversible covalent bond, sharing pairs of electrons between atoms, with D and MTs, DNA methyltransferase, after being incorporated in DNA. Hypermethylation requires that the cell's growth starts after the D and MT inhibition. HDAC, histone deacetylase inhibitors, target the catalytic domain of HDACs, interfering with their substance recognition. Figure 7 shows the effects of these two drugs. Through this video, you can see the many ways epigenetic changes can occur, and the studies support the effect on these changes on animals and potentially humans. Everyone believes that we carry disease or bad genes from our parents in the form of DNA, but through epigenetics we are shown that it is more than just DNA that we inherit. Our parents' chromosomes only account for 50% of DNA, whilst the other 50% is made up of the protein molecules that potentially carry epigenetic marks. Studies shown are just a demonstration of epigenetics and how diet, behaviour, and surroundings can affect generations to come. There is still so much more research for us to fully understand epigenetics, and it is scary to know that this information is just the beginning of it. So what do you believe? Will you choose to change your lifestyle in a bid to better generations to come?